Right, so we're going to start up our next section now and take a look at some DNA structure and also how DNA does what it does as far as telling our cells how to make proteins. So this chapter is all about DNA, what it is, what it looks like, and what it does. All right, so we're going to start off taking a look at the overall basic structure of DNA chemically. We're going to take a look at how DNA stores the information that it does, and from there we're going to go into a discussion of what we call gene expression, which is how transcription and translation pair together as processes to go from DNA first to RNA, and then from RNA to protein. And then eventually take a look at some protein synthesis regulation and also what happens when DNA goes wrong or we have mutations and what kinds of effects that can have on the overall outcome in the organism. Right, so the way we know what we know about DNA is based on several kind of old historical studies that were done. The picture on the left actually shows us what was called, it's just now called the Griffith experiments. It's carried out by a man in his research lab called Frederick Griffith. And what he worked with was a type of bacterium called Streptococcus pneumonia. And what he would do was he would take these bacteria, two different strains. One was called an R strain, and it was called an R strain. R stands for rough, because when these bacteria would grow on a colony, they would sort of form a rough, kind of bumpy surface as a colony. He would then take also an S strain bacteria, S for smooth, because when these bacteria grow on a plate, they created a smooth structure with a colony. So it turns out the rough strain is non-pathogenic, and if you were to inject this into a mouse, the mouse stays perfectly healthy. This bacteria does not have the ability to cause disease, does not have the ability to cause death in the host. The S strain bacteria, on the other hand, does have some pathogenicity to it, so if you inject it into a mouse, the mouse gets sick, and within a couple of days, it's going to go dead. Now, the interesting things are what happens down here. So if we take the heat-killed S, remember S is the one that's harmful, take the heat-killed S bacteria, inject those into a mouse, the mouse is healthy, even though this was the pathogenic bacteria because the bacteria has been killed. Now, the most interesting one of all was when you take the R strain bacteria, the harmless ones that are alive, and the heat-killed S strain bacteria, the harmful ones, mix together living and dead, and you create what's called a transformation of R bacteria into S bacteria. And those S bacteria do have the ability to cause disease and do cause death in the mice. So what we've done here, what Griffith did initially, was to take these bacteria with this green polysaccharide coat around them and transform the ones that don't have that coat into a type that does have the coat, and that coat is actually what makes them pathogenic and allows them to kill their host. Right, so that's kind of a long way around of saying that these bacteria are changing each other into forms that have different traits from what they started off as. The picture on the right is an example, or it's kind of a depiction of the Hershey-Chase experiment, which shows us, again, a different type of transformation, in this case not of bacteria, but in a way of viruses. And so the idea here was that people for a long, long time have known about DNA, and they've known about protein. But what they didn't know up until the 1920s through about the 1950s was whether it was DNA or RNA as the stuff of heredity. Which of those two things actually gets passed on to the offspring to tell those offspring how to create themselves, how to build themselves, how to create proteins and do all kinds of things that cells have to do. So what Hershey-Chase did was it took viruses, bacteriophage, or viruses which cause infection in bacteria. They took some viruses and they labeled them with radioactive protein. So they took sulfur, they labeled the protein with radioactive sulfur, they allowed these viruses to inject bacteria for a while, then they look at the inside of the bacteria to see if there's any radioactive sulfur inside the bacteria. And it turns out there wasn't, because the sulfur is just the protein part of the virus, the bacteriophage. It doesn't actually go into the host, doesn't actually cause transformation. The bottom line over here shows us bacteriophage viruses labeled with radioactive phosphorus. Now, phosphorus is part of DNA. So if you expose these viruses to radioactive phosphorus, you are labeling their DNA radioactively, you allow them to infect their bacterial hosts, and then examine the bacteria for traces of radioactivity, and sure enough, you find out these bacteria growing in the culture do have radioactive phosphorus as part of them. So what that's telling us then is the viruses are actually injecting their DNA into the bacteria, not their proteins. All right,